Hello, DJGuerra here, coming to you from VerEvMed Studios in the lovely American Northwest. VerEvMed, where scientists communicate with people from various branches of other scientific communities, medicine, pharmacy, nutrition, as well as clients from all walks of life to discuss science published in the literature and bring that science to you directly. So we are scientists verifying published evidence in biochemical, physiological, and pharmacological, as well as medical sciences. That's what we do. And these short lectures are meant to give you an idea of the sort of depth that we like to eventually move into when we dig into the literature. So this is an arc of lectures that I've been starting on aging. And we started out with a propedeutic lecture on the epistemology and metaphysics of studying aging. That is, by looking at whether or not aging can be something that is reducible to scientific examination. And then if you go back and you look at the epistemology of aging, the study of the epistemology of aging, one of my previous VerEvMed lectures on YouTube, you'll see that we came to the conclusion that, well, maybe yes, maybe no. So that is what, as far as we can get, is an equivocation as to whether or not using the square of opposition, which involves categorical logic, whether or not aging is really something that is trackable or tractable to study in science. Obviously, we're going to proceed to study aging because it's fundamentally important to humans and to all aspects of life. So we're going to do it anyway. But it's a good idea to think about whether or not reasoning supports the final summary of belief and conclusion that we should be studying it. And that's why that was there. The first lecture in this arc was an introduction of aging. And we went through just what aging is, talked about how old things can get, the orders of magnitude of aging from different living organisms. Today, we're going to actually start into looking at the literature, and we're going to start looking just very at the surface level, the mechanisms whereby aging can progress. And of course, it's going to involve a discussion of the genome. And the genome, of course, is all of the DNA and chromatin that's in the nucleus of a eukaryotic cell inside the human that encompasses the heritable material that allows for the generation of an entire individual. The genome is basically like the base camp where all the genes are, but also the proteins that are necessary to support gene expression. By that, I mean the production of proteins. And those proteins then work in the cell, of course, to carry out all of the metabolic and other architectural necessary pathways so that the cells can function within given tissues, those tissues and those organs within the body. So we have the genome we talk about, which is a strict discussion and look into the genes and their structure. But we also, later on, we're going to talk about the transcriptome, which involves the messenger RNA made from the genome, the proteome, the protein is translated from that messenger RNA, and then a whole host of other omes, such as the metabolome or the lipidome. And those are going to look more discreetly at specific what, where, when, and how aspects of given biological systems. Okay, so that's what omics is about. The ome is what's there, where does it occur, how does it happen? That is, what are the mechanisms whereby it occurs? And then a discussion of why this particular mechanism and not another, and why, when things go wrong, those mechanisms either fail or do indeed support a longevity of the cell. So it's a very brief overview of what omics is. So anyways, let's start off with this really pretty picture. Again, this is in the spring I'm giving this lecture. My name is Dr. Daniel John Guerra, and this is VerEvMed. So right away, let's ask something about aging relative to the genome, which I just described to you. Genome instability has actually been implicated as the main causal factor in aging. So we know that somatic cells are just those that are not germline cells, gametes, that is. So somatic cells, all the cells in your body, are continuously exposed to various sources of DNA damage. Anything from reactive oxygen, such as superoxide or the hydroxyl radical, to ultraviolet radiation just coming from the sun, to a whole host of environmental mutagens. And by those, I don't just mean anthropogenic mutagens, that is, ones made by man. I mean all the environmental mutagens, toxins that are out there in the environment, of which there are probably on the order of billions and billions of different toxic compounds that can affect genome stability. And again, genome, again, is the entire collection of DNA and accompanying proteins that serve to cause inheritance of all of our traits. That is our phenotype. So if you have a genome that becomes unstable, that can mean a lot of things at the level of physiology and even more at the level of biochemistry. So right now, we're just going to look at a 25,000 foot overview on the top of the mountain. And then we'll get into some detail as we go into subsequent lectures. So once again, in response to many thousands of physical chemical lesions introduced into the genome of a typical cell every day, and this has actually been quantified to be on the order of thousands or tens of thousands, there is a highly organized and complex network of genome maintenance mechanisms. And these mechanisms systematically remove damaged chromatin and restore the original structure as close as possible. And not only that original chromatin structure, but all the cohering nucleotide sequences. Remember that the DNA is in a sequence. You have 4 nucleotides. Those 4 nucleotides are non-randomly sequenced so that a 3 of each of those sets of nucleotides will ultimately be translated to an amino acid, and that amino acid will be incorporated into a protein, and that protein will then again serve as the functioning of the entire cell's repertoire of needs. So nucleotide sequence must have high fidelity, almost 100%. There's some wobble there that we can get into later with some other more basic lecture. But for right now, we want to keep that nucleotide sequence as high as fidelity as possible. We also want to keep chromatin structure with high fidelity. So let's take a look at this slide, one I just made. You start off with the original chromatin, and this is just supposed to be DNA and protein inside the nucleus, highly compressed, compacted chromatin. Some kind of damage occurs. There's your red arrow. Now you've got damaged chromatin, and I've shown here by these yellow lesions within the chromatin. And these are probably randomly organized and associated within the chromatin, although there is some suggested that some kind of damaging agents will target specific aspects or locations, loci of chromatin. Anyways, it's just to consider this general overview that we've got damaged chromatin. The repair system kicks in, and when it works correctly, we get repaired chromatin. We're back in the nice green zone. We're back to having our genome spared from any further vagaries, and we have no further mutations, or no mutations are going to be carried on, and that's somatic tissue subsequent to division, cell division, that is mitosis. But sometimes the damaged chromatin is actually either not repaired because there's some fault in the repair mechanism, or it's repaired indeed, but it's repaired in a way that either maintains the mutation or it actually exacerbates the mutation. So repair mechanisms sometimes can cause more problems, and then you end up with mutated chromatin. And the mutated chromatin then is going to give you cells which are going to be carrying that mutation. Again, that's all involved in what we call instability, right? So that instability then could lead to that cell going through various subsequent pathways, such as termination of the cell, that is, killing itself or allowing itself to be killed by outside cellular agents, such as the immune response. And that, an accumulation over time, may be causal or at least highly correlated to what we call cellular senescence, aka aging. So questions you can ask are the severity and or the frequency, so how bad and how often of these, I'm calling them iatrogenic mutations. That means mutations that are caused by the mechanisms that's supposed to heal, right? So iatrogenic means that it's coming from a system that's supposed to repair, but that system itself can introduce mutations. That's what we're talking about here. Are they sufficient to promote or even accelerate the rate or extent by that I mean passing to other cell lineages, aging? After all, we can step back and we already know that cancer itself is an age-related disease. The older you get, the more likely you can get different forms of cancers. And they're clearly manifest. Cancers clearly manifest with an acquisition and metastasis of various chromosomal mutations in what we call epimutations. That is not so much about the sequence of the DNA, but the way the DNA itself is organized within the chromatin and vis-a-vis how it's expressed. Okay, so all of that involves, again, chromosomal damage. Chromosomal damage then leads up, going up the arc of complexity to genome instability, right? And then there's that arc come back down and talk about the phenotype that is the aging process itself. So where are we at? Genome instability is the natural tendency of any genome to undergo alterations under physiological conditions. So any alteration, just by definition, is going to make that structure in this system, the structure we're talking about, the genome, unstable. So just like if you have a broken window, that window's broken, but while you're fixing the window, maybe it's just a small crack in the window, you have to take the window apart, you have to pull it out of the frame, then you have to replace it with a new piece of glass. So in that process of repair, you've actually introduced a much larger lesion, right? You no longer have a window there in the wall. That is, the glass that made up that window is gone as you're repairing it. So that's what we're talking about. The process, the mechanism by which you repair it itself is a natural thing. And what we want to know is that even though that's natural, even though genome instability is natural, that is, it's a physiological process, is it something that just by itself, under normal physiological conditions, nothing really truly pathological, is that sufficient to cause what we call aging? Okay, so let's get more discreet here and ask, what does it mean to say we have a genome alteration? Well, there are at least three ways of looking at it. And we've already sort of looked at these, right? There's chemical damage to the DNA, that is direct chemical damage, and we'll tell you in a moment what we mean in a little bit more detail. But this more generally on a day-to-day basis is considered associated with reactive oxygen species. We call those ROS. Reactive oxygen is normally and spontaneously generated from molecular oxygen in the air. So we're what we call aerobic organisms, right? We use molecular oxygen. And in a previous lecture, I described how molecular oxygen gets reduced to water and driving those electrons into oxygen to make H2O, which is what that is. In so doing that, we make ATP, which is the energy currency of the cell, right? So this is normal oxidative phosphorylation. It's what molecular oxygen is involved in, right? Making energy. Okay, so molecular oxygen is everywhere. It's 21% of the oxygen we're breathing right now. But molecular oxygen, because of its high reactivity and the fact that it is a biradical, that is, it has two unpaired electrons, because of that chemistry, oxygen is always undergoing partial reduction. And that partial reduction leads to partially reduced oxygen species, and that's what ROS are. And some of those are really dangerous to things like the genome. Okay, I'm going to go into detail about reactive oxygen and how it's made and how it's quenched in a subsequent lecture. But for now, that's adequate for us to bring forward. So that's what I mean by chemical damage. It's not just ROS, but that's a major everyday occurrence. And there's nothing to do with living a bad lifestyle. It's just oxygen gets partially reduced in all cells and everywhere on the planet. Second type of genome alteration is a strict sensu stricto mutation. Okay, and this would involve an addition, a deletion, or a substitution of a given nucleotide in the nucleotide sequence. So this is what we think about as geneticists when we talk about mutations. So obviously, if we alter the sequence of nucleotides, depending on how frequently we do that and where we do that, we can alter then ultimately what the messenger RNA looks like, right? We'll have a different sequence in the messenger RNA. And from that, we might have a totally different protein, or even a protein that is so flummoxed it doesn't get expressed. That is, it gets immediately degraded. Obviously, the cell can't go along making a lot of faulty proteins and then getting rid of them, because sooner or later it'll catch up with you. Sooner or later, the proteins would have been necessary are no longer there, or not made in the correct structural form, right? The protein's not made correctly. And that, of course, would lead to total decline of the system. And that decline, after all, is kind of what we look at when we see senescence. When we get into the molecularity of senescence, hopefully we'll be able to totally disambiguate that aspect of it. So third kind of alteration then is what we call an epimutation. And those are heritable, and they are covalent. That means you make a covalent bond, carbon-carbon bond usually, sometimes with carbon-oxygen bond. And so that covalent change that happens to chromatin itself in either the DNA of the chromatin or in the protein moieties, that kind of modification, which normally would control gene expression, now has been modified. And that modification of gene expression, that is, when the genes are expressed, how much transcript is made, and whether or not that transcript is made in the right cell type at the right time upon, say, a spontaneous incursion of, let's say, a stress phenomenon, all of that epimutation ultimately can also lead to total destruction, total dismantling of the cellular machinery necessary to maintain life on a status quo, right? And once you lose homeostasis, that instability that is introduced there, that can then ultimately lead to decline of the cell, decline of the cells within the tissue, decline of the organ, and then you're talking about decline of the individual themselves. Okay, so those are the three possible alterations. Those are general alterations. So let's go into a little bit more detail about DNA damage, repair, and mutations. Only a little bit of detail. So, as I've written here, DNA damage is defined as a deviation from the normal chemical structure of DNA, as distinct from what we were just talking about, mutations such as alteration in the information content, right? The nucleotide sequence. So what kinds of DNA damage are we talking about in terms of just basic chemistry? We're talking about spontaneous hydrolysis. And this can cause things like depurination and depyrimidization. So what are those two chemical processes? When you remove a pyrimidine, that is two forms of nucleotide, or you remove a purine, that would be then depyrimidination or depurination, respectively. What that can cause then is a mutation, right? And that mutation then can ultimately lead to one of those other sequence signatures we just talked about. Or in fact, it can just cause an alteration in the chromatin structure itself. That is the physicochemical structure. Not so much really considering the sequence phenomena, but just the structure of the DNA itself. And then that kind of damage then can lead to lots of other damages to the chromatin. So once you introduce damage, as we talked about just a few moments ago, the repair mechanisms kick in, right? And when they kick in, you can then generate other lesions because the repair mechanisms themselves doing their iatrogenic phenomena can sometimes introduce more mutations. So again, what causes this direct chemical damage? All kinds of things, not things that we can avoid. So radiation from the sun, of course, and as well as ionizing radiation. And there are sources of that everywhere on the planet. And then there are a host of, again, as I said, environmental mutagens, which are not man-made so much as they are just naturally occurring in the environment. They come from plants, they come from microbes, they come from animals, and they come from abiotic phenomena in the soil and in the air. So there are all kinds of potential environmentally generated chemicals which can cause this sort of DNA damage. Okay? That's why you have a really active and robust DNA damage repair system. So if that's not quickly repaired, what you get then is actual DNA damage. That DNA damage, like we saw on the slide where I showed you, could lead then to an actual mutated chromatin, right? Damaged chromatin then, which can then lead to the downfall of the cell and then ultimately maybe enhance the rate or severity of senescence, aka aging. So DNA repair mechanisms themselves are more generally referred to as a means of genome maintenance, right? So we're talking about just maintaining. We're not talking about going in and excavating and starting all over again. We're talking about maintenance of the genome. There are highly conserved systems, as you might guess, that is at the biochemical level, and they're continuously monitoring or surveilling the genome, right? They're looking for any kind of DNA damage or proteins associated with DNA in the chromatin. And they are responsible for that high level of genome integrity that's necessary under physiological conditions, right? So we've got that whole system in place. And again, does that system introduce its own mutations, or does that system become faulty during aging because of errors maybe in the expression of the genes that code for the proteins that ultimately are involved in all that DNA damage repair? And there are probably 100 different proteins involved in that system. Any mutation in any one of those proteins could ultimately decline in the whole system. Decline in the whole system obviously then could lead to a process whereby cells would senesce in order to control further mutation. And then that senescence ultimately can be associated directly with what we call the ambient understanding of physiological aging. Right? So let's summarize here, because we're almost finished with this short lecture. So there is evidence in the literature that chromatin damage and or modification can procure an unstable structure that may trigger a cascade of events that lead to cellular senescence and thus aging. Okay? There is evidence in the literature, particularly when studying cells and when studying model animal systems and model plant systems, model microbial systems, but also various kinds of evidences from the biomedical literature looking at human studies. However, even though that is the case, senescence, which is essentially a result of the failure of DNA damage repair, okay, you can look at it that way, failure, or maybe actually the final end note of what that repair can end up with if it can't do any more. Okay, so there's two ways of looking at what senescence ultimately brings. But senescence actually can spare cellular collapse because it doesn't mean the death of the cells directly, right? So it can abort that cellular collapse, and it also can moderate, right, chronic inflammation. So it's a whole other thing we haven't brought into today's discussion, but chronic inflammation certainly is a major cause of cell morbidity. And that morbidity, if we don't check cellular collapse or moderate chronic inflammation, it can ultimately elevate morbidity to mortality of the individual organism. And isn't that indeed what we're talking about with age-associated illnesses, and age-associated illnesses ultimately leading to the end of lifespan, right? Okay. So I think we can argue somewhat satisfactorily at this point that we need more of a conceptual perspective on the mechanisms of healthy versus pathological aging. So right now, I'm just basically telling you about the how and the what, and not even so much, but a little bit about the when of how the cell controls genome stability. But that's all pretty much at the healthy end. What about at the pathological end? What about when there is an actual damage caused by an agent or by spontaneous cancer? So those two things have to be considered separately and then brought together, both healthy and pathological aging, right? So we need to be able to get an idea about the mechanisms for that before we actually seek to verify what already is evidence in the literature, okay? Because until we understand those underlying mechanisms, it would be more difficult for us to be able to verify and therefore include as truths within our knowledge base, where knowledge is justified true belief, whether or not that evidence does really purport to explain the aging process. So we need to get deeper into it, and that's what we're going to do next time on VerEvMed. So the next time we meet, we're going to dig deeper into those mechanisms, get into some stuff that I like to talk about, biochemistry and molecular mechanisms, but always with the eye on how that perturbs genome stability, and then how genome stability then ultimately is linked to senescence, aka aging. So there's our email address. You can email me at that address anytime. There's our website. We encourage you to visit our website and see what we have to offer and on offer to our clients. Remember that what VerEvMed is scientists verifying published evidence in various disciplines. Here I've listed a few: biochemical, physiological, and pharmacological, as well as medical, biomedical sciences. So thanks a whole lot for your attention. And next time we meet, again, we're going to dig a little bit deeper. Hope you have a pleasant day. And I wish you all extent of a healthy lifespan, at least until next time VerEv meets.